Hello everyone, in this class we'll try to learn how to determine packed cell volume. The aim of this experiment is to determine the packed cell volume. The principle is the RBCs are heavier than plasma. And when oxalated blood is centrifuged in a graduated tube, the cells settle down in the tube. That is its principle. Apparatus which are required are Winthrop's tube, a narrow thick wall tube of length 110 mm, diameter of bore is 3 mm. Graduations are usually from 0 to 10 centimeter, that is 100 mm, both in ascending and descending order in two sides of the tube. The scale with the markings of 0 to 10 from below upwards is used for hematocrit, and the scale markings 0 to 10 from above downwards is used for ESR. Then you require Pasteur pipette, which is a long glass tube of 22 centimeter length with a long thin nozzle about 13 centimeters in length. It is used to transfer blood from the container into the Winthrop's tube. So then you require centrifuge machine, then appropriate anticoagulant, normally potassium ammonium oxalate 3:2 is utilized for this purpose. Then procedure is with the aseptic precaution, draw the blood and transfer it in the bulb containing anticoagulant. Mix the content by rotating the bulb between the palms. Transfer the blood into the Winthrop's tube with the help of Pasteur pipette up to the mark of 100 ml. Avoid air bubbles. Then centrifuge it at around 300 revolutions per minute for 30 minutes. Then record the volume of packed cell and plasma. Repeat the centrifugation till the two consecutive readings are the same. Then multiply the value by 10, which gives you the volume for 100 ml of blood. It is the volume of red cells expressed as a percentage of total volume of blood. Normal PCV is 45% and plasma constitutes around 55%. Note the white buffy coat on the top of the packed cell is due to the WBCs and platelets. Okay. So this is how it looks when you centrifuge the blood, which the remnant, the supernatant remnant is called as plasma, and the RBCs which are precipitated rather settled in the bottom, it is called as hematocrit or packed cell volume, so which is around 45%. And the central, the supernatant is called as buffy coat, which consists of mainly the white blood cells and platelets. So then the packed cell volume has to be reported like packed cell volume of the given sample of the blood is so many percent. If you see the plasma, it has water, proteins, nutrients, hormones, and etc. Whereas buffy coat consists of white blood cells and platelets, where hematocrit is consisting of only the RBCs. In anemia, because of the RBC content is less, the hematocrit value will be less. Okay. In case of polycythemia, because of the RBC content is high, RBC count is high, the hematocrit levels are elevated. Normally in adult males, the PCV is 46% and it ranges from 40 to 50%, and in adult female it is 40 to little less than compared to the males, that is the range is 37 to 47%. So this is in brief about how you determine packed cell volume. Thank you.